Hello, my name is David Glover. I've been asked quite a bit recently, as you'd expect, about the effect that UV light or UVGI has on the COVID-19 virus. So I produced this short video really to explain in very simplistic terms the science behind how UV light affects COVID-19 or the virus which causes COVID-19 and the susceptibility of this virus. Now basically COVID-19 is a respiratory disease which is caused by SARS-CoV-2 virus, and the research that has been carried out suggests that coronaviruses are highly susceptible to UV light. Now what is a coronavirus? Well, they're also known as CoVs. They're primarily found in rodents, in bats and in birds. And they've actually been known, though, since the turn of the century to cross the species barrier. So SARS in 2003 and more recently MERS, the Middle Eastern Respiratory Syndrome, in 2012. The particle itself is a single-stranded RNA virus and surrounded by the corona-like helical envelope. And coronaviruses generally have a size range of 60 to 140 nanometers. So how does UVGI work? Well, ultraviolet light in the C-band attacks nucleic acids. It essentially causes the nucleic acid molecule to form dimers in itself. And what this essentially does is it prevents the molecule replicating, and therefore it prevents microbes proliferating. But really the UVC dosage is critical, it's absolutely critical. But luckily ultraviolet germicidal irradiation, UVGI, UVC, however you want to call it, has been tested on a wide range of microorganisms. When we're designing a system, it's really important to understand the UVC dosage that we're trying to generate to kill the microbe. This is a UV kill dosage. It's also important to understand the amount of ultraviolet irradiation that the lamps, the source of the UV is producing. And as you can see, essentially there's a relationship between the UVC kill dosage, the UV irradiation, and also the exposure time. And what this simple formula is telling you is that relatively low intensity UV light can be used for surface treatment because the treatment time, the contact time or the exposure time of UV light is over a long period. But air disinfection, it requires a high intensity UVC light because the contact time is very short. I've put together a league table, if you like, just as an example of various microbes and the UVC kill dosage that's required. So you can see here on this table that things like Aspergillius niger, that's got a very high UVC resistance compared to the Shigella at the very bottom, which causes dysentery. Which has got a very, very high susceptibility. And you can see here that the coronaviruses in general have a sort of medium range susceptibility to UV. So they are highly susceptible. So when we're designing a solution, the kind of information that we need to get us started really is for a surface treatment, simply we need the height and width of the surface that needs to be treated. And also we need the available distance from that surface so that we can calculate the correct amount of UV light, the correct number of lamps which are going to be utilised. If there's not much space there, then basically we're going to need more lamps because we're unable to get that UV cone of light to fire onto the surface. In terms of air treatment, particularly if you're looking at introducing UV into air handling units for infection control, the basic information we really need is the height, width and available length inside an air handling unit so that we can actually look at what available kill zone effectively we've got for the UV light. And we also need the flow rate. So with those bits of information, height, width and length, as well as a flow rate, we can start to design a simple system for infection control. So I hope that's been a useful sort of overview describing how UVC light works, how coronaviruses are susceptible to UV, and some of the design considerations when looking at infection control solutions. So please feel free to contact me if you'd like any further information. Thank you.